Good afternoon, everyone. Thank you for joining us today, and welcome to the 7th edition of the GSU MalariaGEN Journal Club monthly series. So we run these webinars on a monthly basis, or try to do so. And today's webinar is a vector-focused one based on the newly minted PhD, Dr. Sanjay Nagi's recent preprint titled Parallel Evolution in Mosquito Vectors. A duplicated esterase locus is associated with resistance to pirimiphos-methyl in Anopheles gambiae. My name is Pablo Chauke, and I'm a training coordinator at MalariaGEN GSU, working with an awesome team of people to organize these monthly events. Just a few ground rules. The Journal Club is being recorded and will be available on our MalariaGEN YouTube channel later today. So please do check it out and rewatch and share with your networks. Please stay on mute throughout the presentation. We will have a Q&A session at the end where you can raise your hand or type your questions or comments in the Q&A as well as the chat box. So please, while Dr. Sanjay is presenting, do type your questions and comments in the chat and we'll address them at the end of the presentation. Without further ado, I would like to introduce our speaker for today, Dr. Sanjay Nagi, who emphatically agreed to give this Journal Club. It's always been, I've known Dr. Sanjay for the past 3 years now as a teaching assistant, as a researcher, as a presenter, as a scientist. So it's always a pleasure to listen to him speak. So I'm hoping that today is going to be the same, and I know it is going to be the same. The title of the talk, as you can see on the screen, is similar to the paper that was recently, or rather preprint, that was recently published. I'm just going to read a quick bio for Dr. Nagi and then let him do the presentation. Dr. Sanjay Nagi is a postdoctoral fellow studying genomics of the major malaria mosquito, Anopheles gambiae, at the Liverpool School of Tropical Medicine. A major focus of his research is the rapid evolution and spread of resistance in the major malaria vector, and how we can use population genomics to ultimately inform malaria control programs. Sanjay enjoys developing open source computational tools that can aid the community and empower researchers to analyze their own data. Dr. Nagi, thank you so much for joining us. You may begin your talk.
Thanks very much, Pablo. And yeah, big thank you to everyone at MalariaGEN for inviting me today. I really appreciate it. And thanks to you guys for attending. So today I just wanted to talk about this preprint, which is where we essentially found a new mutation involved in insecticide resistance in Anopheles gambiae. So very briefly, as we may know, since the year 2000, we've made really large reductions in the malaria burden in sub-Saharan Africa. But unfortunately, in the past few years, those reductions have begun to now stall, and malaria is still killing over 500,000 people per year. And so in my department at the Liverpool School of Tropical Medicine, we try to come at this problem from the perspective of the mosquito vector by implementing vector control. And so what do I mean by vector control? I simply mean anything that is targeting the mosquito vector rather than directly targeting the Plasmodium parasite. And so since the year 2000, the majority of those gains that were made can actually be attributed to vector control, either in the form of long-lasting insecticidal bed nets, LLINs, or in indoor residual spraying. So essentially where you spray the insides of houses with a chemical, an insecticide, and so when the mosquito then goes to rest, it will take up an insecticide and die. But unfortunately, of course, so as you notice, both of these methods are based on insecticides, on chemicals. And unfortunately, mosquitoes evolve very rapidly to these insecticidal selection pressures that we exert on them. And this is partly due to the fact that they have extremely large population sizes, and they also have extremely high genetic diversity. And in combination, what this means is you can kind of expect any mutation that you could possibly think of will be somewhere in that mosquito, in that population of Anopheles gambiae. There will be a mutation somewhere. And so we broadly categorize different mechanisms of insecticide resistance into 4 categories. And so there's these 3 sort of physiological ones. So metabolic resistance, in which generally we refer to it is when there's an upregulation or overexpression of enzymes, which eliminate the insecticide. Target site resistance are when you find mutations in the actual target site of an insecticide where the insecticide binds to. Generally, these are proteins in the nervous system. And cuticular resistance generally involves a sort of change or thickening of the insect cuticle, so that the insecticide cannot pass through the cuticle as quickly. And then finally, you have behavioral resistance, which is likely to be very important, but unfortunately, it's something that's very difficult to sort of detect changes to. So this map is just a paper, figure from Penny Hancock's paper in 2020, modeling the spread of insecticide resistance. And this specifically shows the spread of pyrethroid resistance. And so you can see, so pyrethroids are the main class of insecticides, which are found in bed nets. And you can see that in the year 2000, you would find, if you expose mosquitoes to insecticides, you would get a high mortality, which is what we want. But unfortunately, fast forward to 2017 or to 2024, and we have very high levels of pyrethroid resistance in sub-Saharan Africa. So pyrethroid resistance is already widespread, and this is due to the fact that bed nets have been used, at a high level throughout sub-Saharan Africa. But there are now also novel active ingredients for vector control in use. So we now have multiple different types of bed nets, which have mixtures of insecticides in them, and also new indoor residual spray formulations, which do not have pyrethroids but have other compounds such as neonicotinoids or chlorphenapir and so on. And so essentially what we want to do as people who are interested in genomic surveillance is we want to use genomic surveillance to be able to quickly detect any form of resistance to novel active ingredients, so that we may be able to suggest then changes to how we're implementing vector control, such as by using a different active ingredient or different tool. So I just wanted to have a very brief recap on different mechanisms of resistance to insecticides. So in Anopheles gambiae, resistance to pyrethroids, the main compound in bed nets, have been in the past particularly associated with target site mutations in the voltage-gated sodium channel. This is the target site of pyrethroids, and also DDT as well. And there's sort of 2 mutations, L995F and L995S, which were at particularly high frequency. And now we find in Anopheles culexiae, these are actually being replaced by a pair of mutations, V402L and 1527T. Cytochrome P450s, so part of sort of metabolic resistance, have been commonly implicated against pyrethroids. So these include CYP6AA1, CYP9K1, CYP6M2. And generally, when we find resistance based on cytochrome P450s, this is associated with either single nucleoside polymorphisms, so mutations that cause amino acid changes, potentially meaning that that enzyme has sort of enhanced ability to metabolize the insecticide, or also structural variants. So we might find that there are copy number variants, so gene amplifications, with the idea being that that would increase the expression of that P450, or also potentially insertions, which may change the expression of that P450 as well. And then also people have also looked a little bit into cuticular resistance, and it does seem as though that is also playing a role in pyrethroid resistance. In comparison, we can look at resistance to organophosphates. It's another class of insecticide. And organophosphates are not really used in bed nets, but they are the main active ingredient in some IRS formulations. So in particular, there's a IRS formulation called Actelic CS, which has the compound pirimiphos-methyl in it. And this is quite wide. This is a very widely used indoor residual spray formulation that was brought in the year around, I think, 2015 or so. And resistance to organophosphates in Anopheles gambiae have been primarily associated with the target site mutation, ACE1G280S, and also duplications of this gene as well. And so this schematic is just from a paper from an old colleague, Javier, and so he did a big sort of population genomic analysis looking specifically at this locus. And essentially what you find is you either find this is a sort of wild type arrangement where you just have ACE1, no G280S mutation. But what you find often in Anopheles gambiae mosquitoes that are resistant to organophosphates, and this is particularly in West Africa, you find that there has been a duplication of this region. So you have 2 ACE1 copies or more, but some of these copies have this 280S mutation. And we actually know specifically why this occurs. It's because the ACE1 protein with a 280S mutation is actually much worse at its general job than the wild type mutation. However, in the presence of insecticides, it's incredibly beneficial. And so we often find this pairing of a wild type and a resistant allele, which kind of retains the sort of wild type activity, but also gives you some, confers some level of resistance as well. So in the past, a lot of sort of mechanisms were discovered, particularly metabolic mechanisms were discovered with transcriptomics, so either microarrays or RNA sequencing. But we moved towards doing obviously a lot of whole genome sequencing with the Anopheles 1000 Genomes project. And so the Anopheles 1000 Genomes project, as many of you will know, essentially was performing whole genome sequencing for enhanced monitoring and surveillance of major malaria vectors in the gambiae species complex. And this has kind of now become the Vector Observatory, which is a global collaboration creating an open access resource of genomic data from surveillance of Anopheles mosquitoes. And so I think VOBS is now around over 15,000 specimens from 26 different countries in sub-Saharan Africa of Anopheles gambiae sensillata. And so when we started this project, essentially the aim of the study was to use AG1000G data to find novel resistance variants in Anopheles gambiae. So to do this, we essentially used public AG1000G data, and then we also integrated some more whole genome sequence data from the Vector Observatory from the GUARD project. And so this project is from the Donnelly-Weekman group, my supervisors, where essentially they perform whole genome sequencing from Anopheles gambiae and Anopheles Culex from both West Africa and also some from East Africa as well. And so overall, we looked at 2,431 individual whole genome sequence mosquitoes from 8 different countries, ranging from 2012 to 2017. And within that, there was about 1,200 specimens that were phenotypes, and I'll go into that a bit more detail later. So I mentioned the aim was essentially to find novel variants. And so the first thing we did was perform genome-wide selection scans to try and find regions of the genome which look like they're under selection. Essentially, the aim here is we want to find regions of the genome where it looks like there is a mutation which is spreading, basically. And so I split the sort of cohorts we had up into different groups. So we had gambiae from 2012 and 2014, and then a sort of later group as well, and the same for Anopheles Culex, and then just one cohort of Anopheles arabiensis, which are all from Tanzania in this case. And so when we do these kind of, we use these statistics, essentially what we're looking for is we apply the statistic in windows, and we're essentially looking for windows which have reduced diversity. So we're kind of looking for windows in which all of our samples, for some reason in this region, or many of our samples in this region, look very similar to each other. And when you find a region like that, generally that's indicative that a mutation has occurred here, has spread through a population. And so because it spread through a population, all of the sort of individuals at that locus look very similar to each other. And so this specifically is a statistic called H12, which we're using. Those of you who've done the MalariaGEN bioinformatics course will be familiar with that. And so one thing that you can notice from these scans is immediately you see, really large signal at the voltage-gated sodium channel, which we expect because we know this is a very important mutation in insecticide resistance, as I mentioned previously. It's a particularly large signal, and it sometimes is a little bit lopsided due to the fact that it's at the end of the chromosome. You sort of have a region of low recombination. So this is we expected to see, and we also see a smaller signal around here. I think you can see my mouse, hopefully, around 25 megabases. This is the RDL gene, which is another target site resistance mutation involved in resistance to dieldrin. But then we also found this other signal at around 28.5 megabases into the 2L chromosomal arm. And it's a relatively broad signal, which kind of indicates that it's sort of potentially might be more recent selection. And so we were kind of interested in this and wanted to have a look in more detail. So we zoomed in on this locus, and essentially what we found was, really directly underneath the peak, the H12 peak signals, we found this pair of alpha esterases, so COE-AE1F and COE-AE2F, that are on opposite strands from each other. There's also a couple of other genes which could be related to resistance, so aldehyde oxidases, and a couple of UGTs which can be involved in sort of second phase of pyrethroid resistance. However, the signals are really directly over the alpha esterases, and I'll kind of explain why we do focus on these. So something that we were then wanted to do is we thought maybe, a sort of good first step in this situation is to try and see, are there orthologs of these genes in other insect species or in other major vectors? And if so, have they come up as being involved in insecticide resistance in the past? So essentially, we perform reciprocal blast hits with Aedes aegypti and with Culex pipiens. Nothing specifically came up with Aedes aegypti. There's they did have orthologs, one-to-one orthologs, I think, but there's no evidence that they were ever being overexpressed much in the literature or involved in resistance at all, really. However, when we looked into Culex pipiens, we found something quite interesting. So essentially, we find, so I've just showed the sort of equivalent locus in Culex pipiens. So this is the orthologous locus. And essentially we find that there are one-to-one orthologs with two genes in Culex pipiens, or Quinca fasciatus. So S3 and S2. And this was immediately quite interesting because this is a known locus for insecticide resistance in Culex pipiens, known as the ester locus or ester superlocus. And so, in fact, in the sort of late 20th century, so from about the year of 1970 to about 2000, these were the most studied insecticide resistance genes in any species, and were really heavily studied. So just a few examples, but really, these were really heavily studied as a sort of textbook example of insecticide resistance, and an example of evolution in a species due to anthropogenic pressures, basically. And essentially what happened, or what was discovered that had happened in this time, was CNVs that covered S2 and S3 conferred resistance to organophosphate insecticides. And this was specifically in larvae. So I think at that time, organophosphates were being used to control Culex pipiens populations, but at the larval stage. And researchers found that these CNVs increase the expression of S2 and S3, and that haplotypes that are containing these CNVs have spread around the range of Culex pipiens, so literally around the world, and sort of competing with each other. So this was immediately obviously very interesting. We were aware of this locus and these genes in the past, and sort of gave an indication that, first of all, that it may well be the alpha esterases that are driving this selection signals, but also that something interesting is happening here. So given that in Culex pipiens, there were CNVs that have been associated with increased gene expression, these two genes, we wanted to have a look at gene expression data in Anopheles gambiae. And for this, we use a tool called AnoExpress, which I and Vicky Ingham have developed, and there's a recent preprint which went up a couple of weeks ago. And essentially, so this is a meta-analysis of all the published transcriptomic studies into insecticide resistance in Anopheles gambiae. So essentially, each dot here is a different experiment from a different place in sub-Saharan Africa, and it just shows the sort of the fold change for each gene. So this would have been, let's say, a resistant strain from a given country compared to a susceptible, of the sort of lab susceptible strain. And essentially, what it showed is that certainly for one COEA1F, you see quite relatively high fold changes in Anopheles gambiae in particular, a little bit more mixed in Anopheles colutei, although there are some samples which show very high fold changes. And sort of, yeah, similar in sort of Arabiensis, that there's a sort of positive median overall. The data for 2F is a little bit less convincing, but probably worth noting that these are all skewed positively. They don't sort of sit exactly at 0. And this is just showing the proportion of experiments that showed either significant overexpression or significant underexpression. You can see that, so there's 8 gambiae experiments in AnoExpress, and actually 7 of them, this gene was overexpressed compared to these susceptible strains. Colutei, a little bit more mixed for both genes, but Arabiensis also generally either insignificant or overexpression. And this is just the preprint that went up. So the next thing we wanted to do was sort of look at the general picture of selection. So one thing, when we perform the genome-wide selection scans and we get that sort of H12 signal, it doesn't really tell us if there's been one mutation that's occurred, which is spreading, or if there's multiple. And so to look at this, we use a method called diplotype clustering. So some of you may be familiar with haplotype clustering. This is essentially a very similar process. So essentially now we've zoomed in on this esterase locus, and essentially and performed hierarchical clustering basically. And each you can imagine many thousands of tiny little columns in this figure. Each column is a different individual sample, colored by species here at least. So you can see that this is a block of Anopheles gambiae and a block of Anopheles colutei, and so on. And just to help sort of interpret this a little bit, I'll show you examples of what it will look like if there is sort of no selective sweep in a region. So I believe, I'm not sure what region this is, but it's a random one which has no evidence of selection. And so essentially this is what you would expect to see if there are no selective sweep regions. On average, each individual sample is quite far from each other, essentially, in terms of the genetic distance between them in this region. In comparison, if we look at a known gene involved in resistance, so CYP9K1, you have a different pattern where actually you get these clusters of samples, for example, along here, and here, and potentially some smaller ones as well. These big clusters where actually they're all identical to each other. So where you get this sort of flat part, and you can see that the y-axis is distance between the samples, in this case, the number of SNPs. So these two samples, or three, or however many, they're all identical. And so that is really indicative that they share a mutation, which is beneficial, and it is spread to all of these samples. So this, and so that is essentially, there is some sort of selective sweep that's ongoing. So you might consider this to be one selective sweep, and this one to be another one, and so on. So going back to our figure, so we can see that there are what looks like multiple potential selective sweeps. We can see a relatively large one here, and other ones here and here. And essentially we've cut this sort of hierarchical clustering tree and labeled these as diplotype clusters. So you can see here 7 sort of called clusters in total. And we've also plotted alongside that, we've had a look at we sort of estimated copy number in the carboxylesterase genes, and plotted that alongside the data. So what we can actually see here is that, particularly in diplotype cluster 3, there seems to be a lot of individuals which have a CNV here, which is potentially interesting, and which we'll talk about that as well. And also in diplotype cluster 5, it's a little bit difficult to see because I think you need to kind of zoom in this plot to see it. But there are also some CNVs associated with this as well, and this is in Anopheles arabiensis. So what we can see is that, okay, there's definitely multiple ongoing selective sweeps in this region, which is interesting. So we also wanted to have a look at amino acid variation. So this is the same plot now, but we have, it may be a little bit difficult to see, so you'll have to sort of take my word for it. But essentially it's the same plot again, but this time we have, for each alpha esterase gene, we sort of plot amino acid sort of genotypes essentially in each individual. And what this allows you to do is, so you can take one diplotype cluster. Let's say look at this one on the left, and you can see, okay, this cluster has the K20R mutation and the G377E mutation. So essentially the aim of this is to try and see, are there any obvious amino acid mutations which look like they could be driving these selective sweeps? And for the most part, the answer is not really. For example, the ones that I just mentioned are basically found across all samples, so they probably wouldn't be a good candidate for that. However, there is kind of one real outlier to that, which is if we look at this cluster in orange and in red, there is this E477V mutation, which does seem to be quite rare outside of those samples, and that could potentially be important. And we note that these are not associated with a CNV as well. So just to look into a bit more detail, I mentioned that we estimated copy number variation at the alpha esterases. Essentially how this is done is, so we take the coverage values across the genome. So this is the number of reads that align to the genome at any given position, and then normalize that. So on average, obviously mosquitoes are diploid organisms like humans. We expect to have a ratio around 2 essentially. However, when we get a gene duplication or amplification or CNV, we see that should increase essentially that ratio. And so these are 2 examples of CNVs that we found in this data. So one which was found in cluster 3, specifically from Ghana and Togo. You can see how the values, the sort of circles here, sort of jump way high, and this is one alpha esterase, and this is the other one. And so the blue line is a HMM prediction of the copy number. So this CNV that's found in cluster 3, I'll just re-show you cluster 3. Cluster 3 is this one, which I said was tightly associated with a CNV seemingly. Seems to just cover the 2 esterases, and then it splits this gene in half, so it causes an extra partial copy of this aldehyde oxidase. In comparison, we also found a CNV in Tanzania, Anopheles and arabiensis. This was diplotype cluster 5. The CNV that we find at this locus is actually much larger. It covers about, I think it's 11 genes in total, but importantly does cover the 2 alpha esterases. Now, something that's quite interesting, particularly about this first CNV, is that there is actually a CNV in Culex pipiens specifically, which has very similar breakpoints. So it cuts this aldehyde oxidase in half, and then just covers the other 2, the 2 esterases, which is just quite interesting, sort of coincidental, and a bit funny how you find something so similar. So this is a schematic now of, well, the locus, but also the duplications. So we have DUP1, which we find in Anopheles gambiae from West Africa, and then DUP2, which covers many genes, that is only found in Anopheles arabiensis from Tanzania. And in our whole data set, they're at relatively lowish frequency, although we have seen that in more recent data from East Africa, DUP2 is actually, in some areas, for example, Tanzania, is actually very high frequency now already. So at this point, we found this mutation, we found these CNVs that are seemingly under selection, and obviously very good candidate for being involved in resistance, given the sort of evidence from Culex pipiens. We then wanted to see if we could try and find if they're associated with any insecticide. And for that, we wanted to integrate some phenotyped whole genome sequence data from the GUARD project. So this was published in Nature Communications last year. So if you're interested in this, please have a read. And essentially what was done here in each location were larval collections were performed and offspring were raised in the lab, and then bioassays performed to obtain distinct resistant and susceptible phenotypes. And then we performed whole genome sequencing on those individuals. And this was done with 2 insecticides, so deltamethrin, as I mentioned, the main compound in bed nets, and pirimiphos-methyl, which is obviously the compound found in Actelic, the IRS formulation. And I mentioned this was done for both West and East Africa. At the point of this analysis, we only had the West African data. And so this was cohorts from Côte d'Ivoire, southern Ghana, Togo, and Benin. And this table just showed the sort of sample sizes of around sort of 50 dead and 50 alive each from each location. So we wanted to see, I mean, first of all, so we had found CNVs, the CNV in some of these populations. Is it associated with being alive or dead? And when we did that, we couldn't test for DUP2 because we didn't have any phenotype data from Tanzania. So we were just testing DUP1 at this point. And we did find that the presence of at least 1 CNV is associated with resistance to pirimiphos-methyl in Anopheles gambiae. So this plot is just showing the odds ratio. And this was also while controlling for the presence of ACE1. So because we knew that ACE1 is so important for resistance to pirimiphos-methyl, essentially we controlled for that to make sure that wasn't having a confounding effect. So it looks as though indeed that this locus is important for organophosphate resistance in Anopheles gambiae as well as in Culex pipiens. So in summary, CNVs at COE AEXF are associated with resistance to pirimiphos-methyl. This is the active ingredient in Culex CS300. It's worth noting that like pirimiphos-methyl has been quite effective since it's been brought in to IRS, and this doesn't necessarily mean that it's no longer effective, but it does mean that we need to sort of continue to perform surveillance of this locus and monitor into the future to make sure this is not hampering vector control efforts. We found that resistance mechanisms often shared between insect taxa, and this is something people have found in the past, and this is another example. And I think this study is a really nice example of how we can use genomic surveillance and population genomics to identify mutations involved in resistance, evaluate them, and also track them in the future as well. So many acknowledgements. Yeah, a big thank you to everyone that was involved in the study. A big thank you to all of the MalariaGEN and Vector Observatory people who were involved in producing the data that we use. Before I finish, I did want to just touch on another preprint that I think went up last week, which actually shows more evidence of esterase-mediated resistance. And so this is actually the data from the Tanzanian sort of side of GARD, so the East African side of GARD, led by Eric Lucas, my colleague. And what he found was that, this is specifically looking at Anopheles arabiensis, he found that most of the selective sweeps in arabiensis do seem to be driven by copy number variants. But another thing that he did find specifically was he found CNVs in another alpha esterase gene cluster, so COE from 2G to 6G. He found CNVs in this region at high frequency and have also found an association with resistance to PM in a population too. So this is kind of more evidence that esterases definitely look as though they might be important in resistance to pirimiphos-methyl. So please do have a read of that as well if you're interested. So yeah, that's all from me. Any questions? Happy to answer. That's all. Thank you.
Thank you so much, Dr. Sanjay. Brilliant presentation and informative as usual.
Thanks.
We now have about 20 minutes for Q&A. So there's a comment from Anonymous. Thanks so much for this masterpiece and congrats for the preprint. There's a comment and question from Muhammad Gaji. I can see on the selection scan figure a huge signal around VGSC in gambiae and colluti, and even stronger than the alpha esterases signals. What is the implication of this in this study? Does that mean that this region could also drive resistance or cross-resistance to pirimiphos? Which mechanism is stronger in these populations? The KDR-based VGSC, the alpha esterase duplications, do you try to check for an additive effect of the KDR and alpha esterase on the resistance phenotypes? Thanks and congrats once again. Sorry, that was a mouthful.
Yeah, no, that's great. Good questions. Thanks, Mohammed. I don't know if I will remember all of that, but I'll give it a go. So, yeah, so the signals at the VGSC, so it's worth noting, so these populations that we're looking at, for the most part, I mean, particularly the gambiae and collutii, which are primarily from West Africa, we know that resistance is very strong in those areas. They will be resistant to multiple insecticides. So the likelihood is that these cohorts that we're looking at are probably highly resistant to pyrethroids, and they may be resistant to pirimiphos-methyl or organophosphate, or there is emerging resistance in some of these cohorts, shall we say. So the fact that we see really large signals at the VGSC doesn't necessarily mean that the VGSC is sort of related to pirimiphos-methyl resistance. Like the signal here, it's obviously, as you say, extremely large, and we know that KDR mutations are at basically a fixation, which will cause a signal like this. So No, I think in this situation, the VGSC mutations, KDR mutations, will be conferring resistance to pyrethroids and DDT. It's more just a case that, these cohorts are resistant to so many, potentially so many multiple insecticides. That's why, basically. And so that's something to be aware of, I think, all the time, is when we do see selection signals, we never know which insecticide necessarily it's conferring, it may be conferring resistance to, or a single sort of loci may be conferring resistance to multiple insecticides potentially. I mean, that's something here we've only been able to look at 2 insecticides, essentially deltamethrin and pirimiphos-methyl, because that was the only thing we had data for. But it's highly possible that this locus could be, I mean, it's likely to be important for resistance to other organophosphates, but whether it could be important for resistance to other things like carbamates or other class of insecticides is entirely possible. I'm just going to have a look at the chat just to see the rest of the question. I don't know if I can see it. Can I?
You have to go to the Q&A.
Oh yeah, okay. Oh, answered. Okay. Does that mean which mechanism? Okay, yeah. So which mechanism is stronger? Difficult to say. I mean, the VGSC mutations have had a long time to spread, obviously. Yeah. And so we obviously, because of what I mentioned about KDR and esterases being separate, we didn't look for an additive effect. We could have looked for an additive effect of ACE1 and alpha esterase CNVs, which we didn't do. You would assume that there will be an additive effect of both together, but we didn't specifically look at that. Thanks, Mohammed.
And there's a comment from Honorine Kabore. Great talk, Sanjay. Thank you.
Thanks, Honorine.
We still have time for questions, so please do raise your hand if you want to speak, or type in the Q&A or chat box, and Sanjay will gladly answer your questions.
And yeah, happy to discuss at any point. If anyone has any questions offline or thinks of something later, please just email me. I'm happy to have a chat and talk about it.
You mind sharing your email with?
Yeah, of course. Yeah, I don't know how best to do that, but I can share it.
We can share it in the chat or.
Okay. Yeah.
Thank
So
you.
I'll do that.
Dr. Vicky Simpson says, "Hi Sanjay, great talk. So many new preprints in such a short time. What is your secret?" Please do share.
What was it? So many new what? Sorry.
Preprints.
Preprints.
Publishing
Okay,
a lot.
I know.
What's
It's all the many years of PhD work that's just been building, and finally some of them are coming to completion now, which is nice.
Thank you. And there's a question now from Rafi Rahman. Excellent presentation, Dr. Sanjay. Do you think about any particular OP markers like we have KDR for PYR? Sorry if my question looks silly.
Did you any particular OP markers that we have? So I think the equivalent is like ACE1, the ACE1 mutation. That is the target. So like the equivalent in pyrethroids, the target site is the voltage-gated sodium channel. We have KDR mutations at that locus. The equivalent in organophosphates, the target site is the ACE1 gene, and we have that mutation I mentioned, the G2ATS mutation is kind of equivalent, and at ACE1 you also get gene duplications as well. So I think that's what your question is, and that's like the kind of equivalent essentially. And ACE1 we know is very strong. So ACE1, I don't know the odds ratios that we get off by heart, but it's very protective. Like if you have that mutation, it confers like a high level of resistance to organophosphates and also carbamates as well. They have the same target site. So that's the kind of equivalent, if that's your question.
Thanks, Sanjay. There's another question from Muhammad Gaji, and I think on behalf of Jonas, very nice presentation. I realize you use whole genome sequencing to confirm the association of CNV to the resistant phenotype. It would be good also to design a simple qPCR to do the same in resource limited labs. Any comments?
Yeah, good question. So I didn't mention that. I should have mentioned it. We haven't designed a qPCR, but we have designed at least a PCR, so like to be run on a gel for this, which is in the preprint. So that could be done. I can't remember why we didn't develop a qPCR, but I'll have a think about that. But yeah, but there is an assay which you can run, which is in the preprint, to sort of detect that CNV, both CNVs actually, both the one in West Africa and the one in Tanzania, to look at if you wanted to try and track the CNV in your samples.
Okay. Dr. Jola Dora says, do you want to speak a little more about the Ano Express, please?
Okay, sure. Yeah, good point, Joel. I probably should have mentioned a bit more detail. So I don't have any slides on, I won't get any slides, but yeah, Ano Express is a recent project that I've been collaborating with Victoria Ingham on. And essentially, we took all of the published studies into insecticide resistance in Anopheles gambiae and phonestus, and sort of reanalyzed them all, essentially. I mentioned transcriptomic studies, so either microarray studies or RNA sequencing. So we reanalyzed them and put that into a single data set and made a Python package called Ano Express. I'm just going to share, I'm just going to go, oh, I've stopped sharing actually, that's not what I wanted to do. One moment. Here we go. So we've made a Python package called Ano Express, where essentially you can, it's kind of a little bit MalariaGEN data like, nowhere near as large at all, but essentially you can like load the transcriptomic data. If you have a gene that you're interested in, you can, let's say you have a gene you think might be important, you can sort of plot the levels of gene expression for that gene in all of the different studies. Hopefully there might be an example here, maybe not. But yeah, so you can kind of just supply these functions, a list of your genes that you're interested in, or even a region of the genome, and it will like plot the gene expression for all of those regions, basically. And so there was, yeah, a recent preprint. I can share that around, or please just get in touch if you're interested. And if you're interested in using Ano Express on your data or for your genes of interest, yeah, just let me know and I can help you on that as well.
Okay. Any last questions or comments? Please raise your hand or type in the chat box. And as you can see in the chat box, Dr. Sanjay's email has been shared. It's sanjay.nagi at lstmmed.ac.uk. You can also follow him on Twitter or follow him on LinkedIn. And I think you can engage further on the research that he's been conducting over the past few years. So please do get in touch with him directly.
Yep.
Sure.
Definitely.
Okay. So if there are no more questions or comments, I'm going to let you go. Thank you so much, everyone, for joining the March webinar. And Dr. Nagi, thank you so much for your brilliant presentation. As usual, we appreciate the information that you share with us, and we learn a lot from you. To all of you, we did record this Journal Club. It's going to be up on YouTube later today, so please do check out the MalariaGEN YouTube channel. The link is shared in the chat as well. And we are going to be advertising the new April webinar in due course. So please do be on a LinkedIn and a Twitter where we're going to be advertising that. Thank you so much, everyone. Have a good day.
Thanks, Pabalo. Thank you, everyone, very much. Have a good day.